we constructed Food Metabolome Repository, which is a searchable website containing untargeted LC-MS data, including unknown peaks information obtained from a large variety of 222 food samples. I chose foods as target of this data resource construction. It was because food contains a large variety of organisms and also contain processed materials such as fermented and cooked ones. So it should contain a large variety of chemicals. Therefore, it should be used not only for food studies, but also used for all research fields related to the chemical compounds. Okay, I'll show you the basic functions of Food Metabolome Repository. So click the food list menu. Then you can see the food items with image photo like this. So I'll select one of them to see the details. In this case, yes, soybean tofu. Tofu is a famous processed food in Japan made from soybean. Okay. This is a positive mode result from tofu. The detected peaks can be seen on the two-dimensional panel by pressing this button. This is the peak table, and in total, 8,726 peaks were detected. The peak table can be sorted by clicking on the table header. The table shows the retention time, M over D value, intensity, adduct ion type, and database search results. As you can see here, there are peaks with no database search results. And so these are the unknowns. In addition, the availability of MS2 and MS3 spectra and the flavonoid prediction results using flavonoid search are shown. You can get the detailed information on the peak by clicking this button. In this case, let's see this peak. Then the peak information page appears. You can see the details of the database search results by clicking this button, and links to the original databases are available. So as you can see here, many isomers are listed. So we have to judge which one is true. For this judgment, the MS spectra can be helpful. You can see the spectral data as values by clicking this button. This peak showed a typical pattern of drug size. In the MS2 analysis, 271 was cleaved as a major fragment. And in the MS3 analysis, the 271 was cleaved into a set of complex fragments. For this peak, a high flavonoid search result was obtained from the MS3 spectrum. You can see the details by clicking this button. Okay, and here, the isoflavones are predicted. Using these results, you can annotate the detected peaks. So next, let's search the similar peak in the other food samples. Click the Search Similar Peak button here. This page is one of the search functions of Food Metabolome Repository. Peaks can be searched with mass values and retention time. By clicking that button, these two values are inserted automatically and the search results were shown. The peaks with similar mass value and retention time were enriched in pulses. Pulses means beans. The details are here, these. The boxes mean the detected peaks, and the number of the box mean the retention time, and the color means the peak intensity. The same peak detection in tofu was also detected in the pulses, beans. But as you can see here, it is also detected in the other vegetables like parsley. But they have a little bit different retention time, so they would be the isomers. Actually, when I checked the MS3 spectrum of them, the patterns were totally different. So they must be the isomers. As you probably noticed, the pork ham is listed here too. You might think why, but it is reasonable that this pork ham contains protein extract from soybean as an additive. So this pig would be very stable under food processing and might be used for a soybean marker, although I don't know if such marker is useful or not. Let's back to the pig information. You can search the spectral data by clicking this button. The pigs with similar spectral pattern can be searched. This is the search spectrum function page of the food metabolome repository. The spectrum data list of the pairs of mass value and intensity. Mass value and intensity was input here, and then the search results are shown. And this is the spectrum of the query, and as you can see here, this spectral pattern appears specifically in the beans and the bean-related food like this. No other vegetables.